It's been so long. Now we are going to learn about types of stem cells on the basis of potency. Totipotent stem cells are capable of forming every cell in the embryo, and in addition, the trophoblast cells, fetal portion of the placenta. Totipotent stem cells. Pluripotent stem cells have the ability to become all the cell types of the embryo except trophoblast, ICM of mammalian blastocyst. Then, multipotent stem cells are stem cells whose commitment is limited to a relatively small subset of all the possible cells of the body. These are usually adult stem cells, multipotent stem cells. Totipotent stem cells, pluripotent stem cells, next multipotent stem cells. Then unipotent stem cells, which are found in particular tissues and are involved in regenerating a particular type of cell. So, unipotent stem cells which are found in particular tissues and are involved in regenerating a particular type of cell, that is unipotent stem cell. Spermatogonia, for instance, are stem cells that give rise only to sperm. Then progenitor cells are not capable of unlimited self-renewal. They have the capacity to divide only a few times before differentiating. They are sometimes called trans-amplifying cells. But unipotent stem cells and progenitor cells have been called lineage restricted cells. Progenitor cells, unipotent stem cells which are found in particular tissues and are involved in regenerating a particular type of cell. Spermatogonia, for instance, are stem cells that give rise only to sperm. Then progenitor cells are not capable of unlimited self-renewal but have the capacity to divide only a few times before differentiating. They are sometimes called transition amplifying cells. Then both unipotent stem cells and progenitor cells have been called lineage restricted cells. Pluripotent stem cell is there, committed stem cell is there, progenitor cell, then differentiated cells. Stem cell concept. Stem cells have two progeny at division. One maintain the stem cell niche by forming another stem cell, and other cell form more committed cell that can differentiate. And criteria for a stem cell to retain its stemness. The continuous proliferating stem cells are housed in compartments called stem cell niches. Stem cell niches regulate stem cell proliferation and differentiation usually by paracrine factors, WNTs, angiopoietin, stem cell factor, and are produced by the niche cells that retain the cells in an uncommitted state. Once the cells leave the niche, the paracrine factors cannot reach them. The cells begin differentiating. Example of stem cell niche: mammalian hair follicle contain melanocyte stem cells. The mammalian hematopoietic niche. The stem cells that reside in the stem cell niche in a proliferative and non-differentiated state by a network of paracrine factors including FGF3 and activating, which increase the proliferation of stem cells and their respective inhibitors BMP4 and follistatin. Then mesenchymal stem cell. Some adult multipotent stem cells have higher degree of plasticity that give rise to bone, cartilage, fat, and muscle lineage. The differentiation of MSCs is predicted by paracrine factors and ECM molecules in stem cell niche. Lamin and ECM component keep MSCs in undifferentiated state. PDGF, TGFB is critical for chondrogenesis. FGF signaling is important for differentiation of MSCs into bone cells. Human MSCs can differentiate according to the elasticity of the surface on which they are placed. If MSCs on soft matrices of collagen coated polyacrylamide, these stem cells differentiate into neurons. A moderately elastic matrix of the same materials cause the same stem cells to become muscle cells, while harder matrices cause the MSCs to produce bone cells. So, establishing the differentiation pathway of stem cells, neuroblasts. Competence. The multipotent cells become neuroblasts from pluripotent cells from the blastula if they are exposed to the appropriate combination of signals. Then specification. Cells receive appropriate signals to become neuroblasts, but neural differentiation pathways are repressed by other signals. Then commitment or determination. Neuroblasts enter neural differentiation pathways and will form neuron even in the absence of inhibitory signals. We were learning about competence. The multipotent cells become neuroblasts from the pluripotent cells of the blast if they are exposed to the appropriate combination of signals. Then specification. Cells receive appropriate signals to become neuroblasts, but neural differentiation pathways are repressed by other signals. Then commitment or determination. Neuroblasts enter neural differentiation pathways and will form neuron even in the presence of inhibitory signals. Then differentiation. The neuroblasts leave the mitotic cycle and express those genes characteristic of neurons. Levels of commitment. The generation of specialized cell types is called differentiation. Before differentiation, cell is committed to a particular fate. Commitment, developmental fate, potency of cell become restricted. The process of commitment can be divided into two stages. Specification, capable of differentiating autonomously even when placed into a petri dish or test tube. Neutral environment. Cell commitment is reversible at this stage of specification. Then determination, capable of differentiating autonomously even when placed into another region of the embryo, non-neural environment. The commitment cell is irreversible. Then autonomous specification. The first mode of commitment is autonomous specification. Here the blastomere inherits a set of transcription factors from the egg cytoplasm, and the transcription factors regulate gene expression, directing the cell into a particular path of development. In other words, cell cytoplasm is not homogeneous, but rather contain different morphogenetic determinants, transcription factors or their mRNAs, which will influence the cell's development. In this type of specification, the cell knows what it is to become very early and without interacting with other cells. Such embryos are called mosaic embryos, with each cell receiving its instructions independently without cell-to-cell interaction. With the four blastomere pairs of the eight-celled embryo are dissociated, each forms the structures it would have formed had it remained in the embryo. Blastomeres in the embryo are committed to early stage of development. Autonomous specification in the early tunicate embryo. And rocks use a hot needle to kill one of the cells in a two-celled frog embryo, result that only right or left half of the larva developed. Fertilized frog egg, cleavage, two-cell stage, hot needle, dead tissue, living tissue, half living tissue. Okay. In case of blastula, then destroyed half and neurula stage, half embryo was formed. Then conditional specification. It is the ability of cells to achieve their respective fates by interactions with other cells. Here cells are specified by paracrine factors secreted by its neighbors. In 1888, August Weismann proposed the first testable model of cell specification, the germplasm theory, in which each cell of the embryo would develop autonomously by each cell received different cytoplasmic determinants from the fertilized egg according to the type of the cell. Conditional specification, regulative development in frog. Conditional specification, what a cell becomes depends upon its position in the embryo. Its fate is determined by interaction with neighboring cells. If cells are removed from the embryo, the remaining cells can regulate and compensate for the missing part. Conditional specification produce regulative embryo. Intact 4-cell sea urchin embryo generates normal Plutz larvae. Isolate 4-cell embryo from normal but smaller larvae. Larvae are not identical but ability to generate complete embryo. This is a demonstration of conditional specification. Normal larva is there, intact, separate into 4 cells, larva development from single cells of 4-cell embryo. Then demonstrated that the potency of an isolated blastomere is greater than the blastomere's respective fate. Sea urchin embryo is an equipotential system. Independent parts together form a single organism. The fate of a cell depends on its location in the embryo. Pressure plate experiment for altering the distribution of nuclei. Sea urchin, autonomous specification, micromere, conditional specification, cells around micromere. Autonomous, micromere. Frog, autonomous specification, head endodermal cells. Conditional specification, brain. Morphogen gradients and cell specification. Throughout this book, we will see many instances of cell fate specification that involve morphogen gradients. A morphogen is a diffusible biochemical molecule that can determine the fate of cell by its concentration. So morphogens can be transcription factors produced within cells, as in Drosophila embryos described in the following section. They can also be paracrine factors that are produced in one group of cells and then travel to another population of cells, specifying the target cell differentially according to the concentration of morphogen. So uncommitted cells are exposed to high concentrations of the morphogen nearest its source of production are specified as one cell type. When the morphogen's contraction drops below a certain threshold, the cells are determined to another fate. When the concentration falls even lower, a cell of the same type, initial uncommitted type, is specified in yet a third manner. Then morphogen gradients provide a very important mechanism for conditional specifications. The specification of the multipotent cells in the tissues is accomplished by threshold concentration of the morphogen. Highest concentration, threshold concentration. Morphogen diffuses from site of synthesis to site of degradation. Then syncytial specification. A cytoplasm that contains many nuclei is called a syncytium, and specification of perceptive cells within such a common cytoplasm is called a syncytial specification. Insect egg cytoplasm is not uniform. Nuclei in the anterior part of the cell will be exposed to cytoplasmic transcription factors that are not present in the posterior part of the cell, and vice versa. The interactions of nuclei and transcription factor result in cell specification take place in a common cytoplasm. Autonomous specification in most invertebrates, conditional specification in vertebrates, and syncytial specification in insects. Autonomous specification by cytoplasmic molecules present in the egg, but conditional specification by interactions between cells. Then syncytial specification by cytoplasmic region interactions. Then autonomous specification, invariant cleavage produce same type cells in each embryos, but in conditional specification, variable cleavage produce no invariant fate to the blastomeres. Then syncytial specification, variable cleavage produce no rigid cell fates for particular nuclei. And first one produce mosaic embryo, next one produce regulative embryo, third one produce both mosaic and regulative.